Okay, today we are introducing our very first lesson, that is inheritance and evolution, which is topic 7, and it's the very first topic in senior 6 term 2, as far as the syllabus is concerned. So that means we are going to first and foremost cover inheritance, then later go to evolution, just like the way the statement goes. So the very first thing we are handling is inheritance. As you know, I always give a chance to anyone who is ready to begin the topic with me. So, who can tell me what inheritance means? You leave the biology. I just want you to tell me what you know about only the word inheritance, the English meaning. Yes, English meaning. Anyone? I don't want you to tell me that inheritance is when one takes over the property of the father. No, that's something different. That's something different. There is some other better English meaning that I would want. Yes, Becky.
So, according to my opinion, I think inheritance, to my own understanding, means passing on of parental characteristics from parents to their offspring. When the offspring inherit the characteristics of the parents, that's what I understand by inheritance.
Exactly. Yeah, the way she has just said it, it's just perfect. It's just perfect. Yeah. So, I've said that we are going to begin with inheritance. Then other things will come later. So the evolution will come in later on. Inheritance. So what we are going to begin with, then evolution will come at the right time. So, lesson targets. So that the lesson targets make sense, let me first tell you this story, then I'll repeat it in writing. Becky has told us that inheritance simply means that we are passing on traits from parents to offsprings. Like we are inheriting, we are taking over the traits our parents had, us as offsprings, us as the daughter cells, something like that. So the thing is, who is help us carry these traits? Because it's a parent, then the parent gives birth, but then you find it that they share some traits. They have some traits in common. That means there is a mode of transfer. So we need to know who exactly is helping us to transfer the traits from the parent to the offsprings. And you all know that is DNA. That's why people always go for DNA tests to really confirm, is there a similarity in our DNA structure? If it's there, that means that that offspring belongs to that exact parent. If there is a difference, then that's a red flag. So that means a similarity in DNA simply means that that offspring exactly came from the parent. And in most of the cases, the traits will be transferred. So you need to know what DNA is. Most of you, when you even just check the internet right now, DNA stands for something, but this part means nucleic acid. The Na means nucleic acid. And that's the first thing we are going to talk about today. So after us looking at the meaning of inheritance and realize it is traits passing on from parents to offsprings, we realize it is DNA that is the mode of transfer. And DNA, just from its full form, Na is for nucleic acids. That means we need to first study the biology of nucleic acids. And that's why we are here today. As simple as that. So one thing I beg is you will allow me the very first lesson target. We shall first understand the meaning of inheritance. Then after the meaning of inheritance, we shall have to introduce nucleic acids. And you're going to learn that DNA is a nucleic acid. Just don't worry, we're going to introduce them very well. So, as you know, biology is always having a lot of content. So if I decide to just write and write everything, we may delay a bit. So I decided to get some good notes. I also don't know where I got them from, so don't ask me for my notes. They are the ones we are going to be using. We're going to be reading together as we explain. We read together, we explain. We read together, we explain. As you take screenshots, then later you can refer. As simple as that, nothing much. So when we introduce nucleic acids, we are going to see how far we can go. And then I think we shall have, since today we are just introducing them, we are going to see where we are going to be in position to stop. And then we have a next stop for lesson 2. Yeah. So that is it. Nothing much, but at least you're supposed to leave this place when you know DNA very well. At least DNA. You should leave today's session when you know everything about DNA. And that will have been a fair game. So that will be the end. Yeah. Introducing nucleic acids, most importantly, their structure. And their structure is very complex. So if by God's grace you get each and every component of the structure of nucleic acids, you'll be good to go. Mostly the structure. Mostly the what? Mostly the structure. So hope you now have a plan. That's the plan we are following. I've given you the roadmap of today's lecture. Okay, now, inheritance. 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 So, we said once again that in biology, when you say that something has been inherited, or when you say inheritance, we are meaning that these parents are simply passing on traits to the offspring. These parents are simply passing on what are they passing on? They are passing on traits. You get? And how do you really confirm that an offspring belongs to that parent? It is usually through DNA tests. You've seen this in real life. So that means it is always DNA that helps us to transfer. So the mode of transfer, actually there is a very common word these days. There are people who always say my border guy, my border guy. Everyone who says my border guy think that they have actually made it in life because they have a border guy. But still that remains public transport. So that means I'm going to come here and say that the border guy for the traits is DNA. Now, I don't know who's writing in our screen. I don't know. That's very bad. So the border guy for these traits, the one who is going to help us transfer them is DNA. Please leave my screen. Is DNA. This is what we said. And I said that don't focus about what D means, even if you check the internet. Just leave it. Just concentrate on the NA. And I told you when you check the internet, just ask the internet, what does NA stand for in DNA? And I told you that means it's standing for nucleic acids. Yeah, nucleic what? Acids. And the mere fact that, so before we even look at how traits are moving from parents to offspring, we need to know who is carrying them, DNA. And what is DNA? It's a nucleic acid. So what are these nucleic acids? They're the ones we are supposed to first look at. So the very first thing we're supposed to do today, what are they? So if we answer that question today, we shall be done. What are they? That's why we are here today. So you now know inheritance. That means we have achieved the first lesson target. And now we are going for the second. Let's introduce them. So just like the way you hear the word nucleic, nucleic simply means that they are found inside the nucleus. There is no rocket science there. Nucleic simply meaning they are found where? Inside the nucleus. Then when they say acid, that means they are acidic in nature. So we need to know who is giving them the acidic nature. And we shall see that in their structure. People, just listen to the biology. With biology, if I decide to write, that means I will live in the future. So that means I request you that even after you attending this live lesson, you get time and do what? And get back and listen to those statements that I'm not going to be writing but mentioning. Like if I may ask, what have I said? Why are they called nucleic and why are they called acids? Who can tell us if you've been listening? And maybe if you're serious enough that you noted down. Who can tell me why did I say but evidence, what is your combination? I see you in physics lessons, biology lessons, chemistry lessons, and math lessons. Yes, Vivian.
Good morning, doctor.
Mm.
Nucleic acids, you said they are called so because they are found in the nucleus.
That is the nucleic part. Then for acids, what did I say? Why are they called
They are,
acids?
they are acidic in nature.
And we shall see that when we look at their structure, what really makes them what? Acidic. So that's a very important statement that I've not mentioned, but very important. We are going to see. Thank you so much. Thank you so much. Now, I want you to look at this. You people, if I may ask, you know what they call amino acids, isn't it?
Mm.
This is just to take you back a bit. This is a take back. Someone tell me, you know amino acids. When two amino acids combine, what do they give me? Two only. Then what if there are three amino acids? Yes, Akrod, you're going to tell me all. Then I'll say.
Yes, good morning, Dr. and the clergy. When we get an amino acid plus another amino acid, we get what we call a dipeptide.
That is very
Then
good.
when
You
they
said dipeptide.
then when they are three, they form what we call a tripeptide. Then when they are more than three, we form what we call a polypeptide.
A polypeptide. So where is the protein?
Yes.
You've had.
A protein itself is a polypeptide.
So dipeptides and tripeptides are non-proteins.
They're also proteins.
Yeah.
Yeah.
Thank you for the try. Very good. So he has told us that proteins are basically polypeptides. Yeah, but that doesn't mean that the rest are not. But at your level, as far as your level is concerned, but even if not your level, generally, yeah, proteins are basically that, but that doesn't mean that the rest are not. And you already see that in digestion in man, though it's not part of your syllabus. So I request people, after you speak, don't forget to mute. That is the rule. Because I waste time when I come back to mute you again after unmuting you. Hope you understand. Okay. So I'm saying that proteins are usually polypeptides. Just wanted you to know that. Most of you told me chemicals of life, we know them very well, we covered them, we understood. So you should be a very good student of chemicals of life to actually connect here. Because this now was taught under what? Chemicals of life. So proteins are basically those. You may ask, why are we really doing this? We are going to connect it a bit to the nucleic acids. Just don't be worried. Just don't be worried. So we say, that's why we usually make a statement and say that amino acids are the building blocks of proteins. Because you can see 2 of them dipeptide, 3 of them tripeptide, and when there are so many, they form a polypeptide, which usually means a protein. That means these ones are the building blocks. That means these ones are the building blocks. And those are the building blocks of proteins. Yeah. Hope you understand when you say building blocks, eh? Yeah. What we have just done is the building. Yeah. Okay. Now, let's now go to our notes and try to read through then connect. Yeah. Here. So you can see, someone may be like, but why are we really starting with these ones? When we go to the outline, according to your syllabus, so this topic is divided into 6 sections. 6 sections. I organized them myself. 6 sections. And you people, you should take the section competences very serious, because that's where questions will come from. All the data analysis they are going to set for you in your biology papers will be focusing on the section competences of the different topics. So if they are to set about the structure of DNA, what will the question be about? They will want you to analyze the structure and function significance of the nucleic acids in meiosis and mitosis. They would want to see their role in cellular functions and how mutations in the nucleotide sequences can contribute to diseases like cancer. So don't think things are going to be obvious, telling you to just describe the structure of DNA. Never. So they will assume that you know the structure very well, and now you come up to break down the structure and be like, what is this one going to help me with? What is this one really going to do? Things like that. So you need to be ready, and you're supposed to take the section competences serious. It's the section competences that are going to be converted into the data analysis scenario items. Hope you understand. So for today, we are going to concentrate on part A. Yes, we're going to concentrate on part A. The 3D models of the DNA and RNA. Yeah. And trust me, when those ones get done, these other things become easy. Because whatever we shall be talking about will be just obvious, obvious, and obvious. Then section 2, it will be more of you people doing research, because it is just about genetic technology or gene technology. It will not be bad. Yeah. Provided you understand this first part. This is the most high yield. The rest is very okay, and it makes total sense if at all you know the basics of inheritance. The rest of the things are just what? Obvious. So why I'm focusing on the nucleic acids, their structure and function, is because of that. Is because of what? That. And when you look at that course outline I usually give you, you know that the very first subtopic is nucleic acid structure and function, period. Okay, so I think I'm going to go back to my book. So let's read together. In case any statement doesn't make sense, you simply do what? Raise up your hand and ask. But I don't think any won't. Okay, so nucleic acids and RNA. What are they saying? They are saying that nucleic acids, so we are beginning from here. Nucleic acids form the genetic material of all living organisms, including the simplest viruses. The term nucleic acids come from the fact that they are found mainly, so you should note that word mainly, because if you studied cytology, you realize that there is DNA we don't find inside the nucleus, the one they call the naked DNA of the prokaryotes. So that's why you're seeing there the word mainly. That's why I told you biology is supposed to listen. Now I've mentioned something important, that prokaryotes have naked DNA. And that's why they are saying that mainly, because not all of it is found in the what? In the nucleus. And the DNA found in the nucleus is what they call the true DNA, or the eukaryotes. So what are they saying? So just like proteins, nucleic acids also have building blocks. And those building blocks are the ones they call nucleotides. So either call them building blocks, or you can call them what? Building units. You understand? And just like amino acids arrange themselves to form extremely long molecules called the polypeptides, the nucleotides arrange themselves to form extremely long molecules called what? Polynucleotides. So I had to take you back a bit to the proteins. I wanted you to relate that proteins had building blocks called amino acids, and on arrangement they formed, or on what we call condensation or repetitive combination, they formed what we call polypeptides. So the nucleic acids we are looking at today have their building blocks known as nucleotides. So you should make sure you have right spellings. You get? So as amino acids give us what? As amino acids give us polypeptides. Polypeptides, you need to know that now the nucleotides, these ones we have said they are the building blocks of the polynucleotides. Of polynucleotides. And guess what? In biology, the polypeptides are the proteins. And then the polynucleotides now are the nucleic what? Acids. You know that people are getting it? Why I wanted to connect the two. Because they tell you amino acids give you proteins. And then we again say they arrange themselves repetitively combine to give you polypeptide. That means polypeptides are the proteins. Then we say the building blocks of nucleic acids are the nucleotides. But when so many of them combine repetitively, they form what they call polynucleotides. That means polynucleotides is the biological name to mean nucleic acids. So when I use the two exchangeably, don't get confused. Hope you are together. So we proceed. When I leave you, you immediately complain and be like, Doctor, you've left me. Now, I don't know whether you people recall the structure of the amino acid. You get? Because that means before you look at the structure of nucleic acids, you're now supposed to first understand what is the what? The structure of a nucleotide. You all know that you cannot understand the structure of the protein unless you know the structure of the amino acid. Who can give me the structure of the amino acid? As a statement, I give you some money. Yes, I want a statement. Yes, Steffi, unmute. I want a statement. Okay, even if it's not a statement, but all the marking points. If you don't miss any marking point, I'm going to give you 100,000. 100,000. Yes, Steffi, start.
Thank you. An amino
Mhm.
acid is a two-carbon compound
Mhm.
composed of an amino end and
Mhm.
a carboxyl end.
Mhm.
Yeah,
Okay.
that's all I could say.
Good try. I'm going to give all people chances, but no one is going to take my money. Vivian. But what Steffi has said is right. But I want someone who is going to complete all the marking points. 100,000. Yes, Vivian.
An amino acid is made up of a carboxyl group,
Mm-hmm.
the amino group,
Mm-hmm.
the central atom, which is the carbon atom,
Mm-hmm.
a hydrogen atom bonded to the carbon atom,
Mm-hmm.
and R, which is bonded to the central group, where R either represents a hydrogen or hydrocarbon chain.
Okay. Okay. You repeat R. Where is R bonded?
On the carbon, central carbon.
Central carbon.
Yes.
Okay. Let me hear from Fahad. Yes. Very good. Very good. I'm going to give you my answer later. I don't want you to say that I
Yes,
did cheat anyone. Yes, Fahad.
yes, good morning, members. Good morning, doctor. The structure of amino acid is that it consists of a central alpha carbon, which is bonded to carboxyl group and amine group, and it's also bonded to
Bonded to
R
carboxyl group and what?
and amine group,
Okay, not bad.
which
Okay.
is also bonded to hydrogen atom plus R, which
Mhm.
represent any other function group.
Mm. Okay, that is also good. I want only 5 people. Yes, praise. I think praise is the third, is the fourth. Yes, praise. Then I think I will end with shalom. Yes, praise. No one is going
Morning.
to take my money.
Other
Mhm.
An amino acid is a compound with a central carbon bonded to the amine group, which gives its amino nature, and a carboxyl group that gives it its acidic nature, with an R group as a side chain, which is either represented by an hydrogen atom or an alkyl group. That's all I can say.
Okay, then shalom. Hey, I'm bringing my answer. Uh-huh.
An amino acid is made up of an amino group and a carboxyl group. The
Mhm.
amino group is attached by a covalent bond to the alpha carbon. Then it also has an R group, which is a side chain, and this R group is different for each of the different amino acids.
Yeah, good. Okay. Now, you people, I want us to now vote. And then see, yeah, those are 5 people. They're the ones I wanted. The rest of you wait for other structure. So let's first have the structure of the amino acid. This is how it looks like. This is how it looks like. There is this carbon, the central, isn't it? Then there is this side where we have the COOH, but this is biology. Even if I don't expose the carbon to oxygen bond, you get? Then what do I have this side? I want you to tell me that, doctor, you give this person their money. That there is nothing like this person tried. I wanted all marking points. The person who was about to take my money was Praise Carlton. Then you all know that we have our H, like that. Okay. Now, let us start. That's how the amino acid looks like. If we want, you can expose the bonds, but that's not necessary for today because our topic today is nucleic acids. But of course, you also need this structure. So what we have here in the middle, this one is what they call the central carbon atom. But the most correct word is alpha, central carbon atom. Yeah. But at your level, they may be like, it's okay. But have it in mind, it's there in biological science, which is your reference textbook. But in most cases, they just, provided you say central carbon atom. But the best word should be alpha. Someone may be like, why alpha? All carbons that come after the functional group, yeah? All carbons that come after the functional group, they are called alpha carbon atoms. I told you about alcohols. Are you seeing this? I told you such things. So why they call it the alpha carbon atoms? Because they are those carbons that are coming after the functional group in most of the cases. But in case alpha is not your thing, you leave it because even only one person said it. That's why even they don't even prioritize it. But that is it. And then this one is just the hydrogen atom. I'm going to tell you where my marks are. Then you tell me who had all the marks. Then this side we have what we call haze. So this is where people were not getting marks. People here, you were only telling me the carboxyl group, but at least I wanted you to tell me its nature. Hey, I was interested in its nature. People, just like the way you talk of this Arab group as being varying, just like Sharon said, that it is a varying Arab group, that it is different for the different amino acids. Okay, why didn't you stop on Arab group? So as you also talk about the carboxyl group, tell us that it is acidic. You get? That's what I wanted. Yeah, so Carlton Praise had tried, but she instead said that, okay, she said the amino group gives it its amino nature, not the basic nature. So I wanted this one to be basic amino group. And you people in biology, they allow you to use diagrams if at all you're not good at statements. Now they will come and mark your diagrams. So what you need to know about now this Arab group is that it is varying. Yeah, varying Arab group, as people say it. Varying Arab what? Arab group. And you all know in case it is hydrogen, our amino acid is glycine. In case it is CH3, you know it is alanine. In case it is in case it is the ethyl, you should know which one is that. In case it's the isopropyl, I taught you isopropyl in chemistry. Understand which one is it. So even if you just stop on varying R group, when you say varying, you're trying to mean different amino acids have a different R. Don't go any further. Yeah, that is enough. So I think you realize that no one had my marks. What did I want? That the amino acid is composed of the alpha central carbon atom, to which an acidic carboxyl group is directly bonded. To which an acidic carboxyl group is directly what? Bonded. And of course, a basic amino group directly bonded. You people, I told you that amines are what? Are weak bases. So when I say basic amino group, it shouldn't confuse you. And you know that the carboxyl group is for carboxylic acids which are acidic. The amino group is for amines which are basic. That's not bad. So as well, we have the hydrogen atom also directly bonded to that central carbon, and also the varying R group also directly what? Bonded. Don't bother about the directly bonded, it's not so important, but provided you name correctly the central carbon atom, then you name correctly the groups that are attached to it. So just like the way we study the structure of the amino acid alone, then we go to the peptide, dipeptide, like that. It's the same way we're going to do for the nucleic acids, which are the polynucleotides. That before we look at how polynucleotides are formed, we should now first understand what is the structure of the nucleotide alone. And after us understanding how the nucleotide alone looks like, we can now understand. Yes, we can now understand the structure of the polynucleotide. But at this moment in time, I believe people know that when you get two nucleotides, you get what they call a dinucleotide. Yeah, because we need that thing so much in biology. Dinucleotide, trinucleotide, and then polynucleotide. Very important chemistry. It is chemistry, hope you are aware. So, back to what we are supposed to look at. Shalom talked of covalent bonds. It's very okay because, of course, the bonds are between non-metals, so they are covalent. And I've been teaching you that in organic chemistry under bonding and structure. Okay, now I want us to now look for the structure of the nucleotide as also another building block. Yeah, this one is a bit serious. Don't think it's like those jokes of amino acids. Structure of the nucleotide. It's what we are going to look at today. This one is a bit serious, but for you, just listen. So the nucleotide is having three components. At least you just recall that the nucleotide is having three components. Three components, and make sure you never forget them. There is a way I always tell students to recall, which is wrong, but it can of course help you recall very well. And I'm also going to give it to you. I'm not going to just list the components, but this is it. If you're having your nucleotide here, and you all know it is the nucleotide that is having three components. You get it? You know that this nucleotide is what will give us the polynucleotide. Of course, this is after repetitive combination, you are aware. Now, some of the examples of the polynucleotides we have DNA, and you all know the other friend of DNA is RNA. I think you've heard of that before. You're not hearing RNA for the first time. These are the nucleic acids that we have. DNA, RNA. I think they talk about it in cytology at some point, but slightly. So, these are polynucleotides, but they are going to help us recall the three structures or the three components of the nucleotide. So, this one, you all know that that D, if you check the internet, okay, this R, when you check the internet, it stands for ribose. Just because your curriculum doesn't have carbohydrates, but that was very bad. So you first understand one thing, that DNA is a relative to RNA. Why? Both are nucleic acids because of the Na. Now we know that Na is for nucleic acids. So the R stands for ribose, but I'll tell you the full name, don't worry. So what do you need to know about this ribose? This ribose is a pentose sugar. I don't know. Do you people get me when I say pentose sugar? Who can tell me the meaning of the word pentose sugar? I told you you're supposed to be a very good student of chemicals of life for you to understand this. Who knows what they call a pentose sugar? I need to test your biology so that as we progress, I'll be knowing how exactly to handle you. Yes, evidence.
Sami, what I know about a pentose sugar, I know it has 5 carbon atoms.
Thank you so
It's a
much.
5 carbon sugar.
That is it. That is it. The moment they say pentose sugar, it simply means it is a 5 carbon sugar. It is a sugar with 5 carbon atoms. Hope you people understand it. So that one, I'm not going to write it. Okay, we are going to come across it, but first understand that it is a pentose sugar. Pentose sugar. So I think, let me try to draw it for people. Let me first try to draw it for people, then we shall go back up, because I don't want you to cram anything. Let me first tell people about this ribose. Then we connect it to deoxyribose. So ribose, we are saying it is a pentose sugar. So in case okay, let me first define and be like pentose sugar, we are saying it is a 5 carbon sugar. 5 carbon sugar unit. Whether they put unit, but all we are interested in are 5 what? 5 carbons. I want you to listen. So that means if I told you you're looking for 3 carbon sugars, they are called triose sugars. You get? When you're looking for 4 carbon sugars, they are called tetrose sugars. When you're looking for 6 carbon sugars, they are called hexose sugars. For us who don't want to write the word sugars, we always call them pentoses, trioses, tetroses, hexoses, and then we leave. So either say triose sugar or trioses, tetrose sugar, tetroses, hexose sugar, hexoses, pentose sugar, pentoses. So when they tell you that pentose sugar, this is how they always look like. So you draw the pentagon. I want you to listen. You draw the what? The pentagon. I don't know if people know how the pentagon looks like. Let me draw it here on top here. You all know that the pentagon looks like this. Like that. Or they can decide to draw it like this. Isn't it? And this is a better structure in biology. So I'll give this a full tick, then this one a half. They usually like that structure in biology more. So and a half, the other one is a better tick. So we are going to use the pentagon to draw this structure. I want you to listen carefully, otherwise you're going to be seeing weird things. So first understand how that structure comes about slowly. So we have said we are going to consider a pentagon. So if I'm to draw my pentagon, you're going to be surprised. It has a certain anomaly, which usually gives people a hard time. So I'm going to come and draw my 5 carbon atoms. I'm going to come and put here my carbon. There, I've told you the better structure. Then I'll come and put here my carbon. I'm going to put here my carbon. Then I'll draw my carbon. Then I'll draw my carbon. Then I'll draw my carbon like that. Yes. Then after, I'll do this as usual. But something fun is happening at that point. So for us, we expect carbon to be there so that the structure is complete. but something else happens. Something else happens. And what is that? We instead have there the oxygen atom. So someone may be like, but you said it is a pentose sugar and the carbons are 5. Where is the fifth carbon? Because now there are only 4. Because the fifth position has been taken up by the what? By the oxygen atom. So it's all simple. The fifth carbon is here. Like that. That's where we are having the 6th carbon. Sorry, the 5th carbon. So you first understand that framework. First understand that one alone. You listen carefully. Are we together up to that point? So you draw the pentagon in the preferred structure, but to make matters worse, this pointed part, instead of having the carbon atom, it's having the oxygen atom. So that makes us to demand for the 5th carbon atom, because now there are only 4. And where does it go? It attaches itself here. So it's projecting outside the ring. You understand? So after that, the next thing is putting the OH groups. That's what usually disturbs here. Now, let me simplify your work. How do you show these OH groups? These carbons, I don't know if you people can see. We are going to come and put OH here. So these carbons, I don't know if you can see. For them, they will have their OH on top like that. They'll be projecting up. Do you understand? Hey, they'll be having their carbons, their OH projecting up like that. Then these other ones, they'll be having their OH going down like this. OH and OH. You need these structures so much so that you don't just cram things that are so obvious. OH, even here OH. Someone may be like, what of this carbon? So this carbon doesn't have any contribution because it gave its bond to the 5th carbon. So it will just have to stay silent, nothing else. So people, that is the hardest part of the structure of ribose. That is the hardest part of the structure of ribose. I don't know if we are together there. So the rest of the bonds, just like in organic chemistry, they are taken up by hydrogen. You can see that this carbon is having only 3 bonds. That means that carbon will need hydrogen. So we shall come and give it the hydrogen. You understand? Then you realize even this carbon will need hydrogen. So you come and show it here. In most cases, they show these hydrogens in a projecting way. So they come and now put the hydrogen down. This is how these structures in biology are best drawn. When OH is up, hydrogen is down like that. Then you shall have another hydrogen like that. You understand? Yeah. Then this carbon is also remaining with only 1 bond. So we shall have the hydrogen also coming down. But then the fifth carbon is a bit so specific that it's having only 2 bonds. So it will need 2 hydrogens like that. So if we are to label, you all know that we are supposed to come from here. This is carbon 1. Yeah. So have it in mind, that is our carbon 1 on these structures. Then this one is carbon 2. Then this one is carbon 3. This one is carbon 4. And this one is carbon 5. You need to understand this, because if you don't, then you will not understand the structure of other things. Remember, this is just a pentose sugar. But remember, we haven't even talked about the structure of a nucleotide. We haven't even talked about the structure of a polynucleotide. We haven't talked about the structure of DNA. So it is kind of much. But if you understand from basics, as we grow the content, it's going to be obvious. So that's the numbering. We number things like this in that direction, as you can see. Now, we can condense this structure. Remember, this is science, and we all know that most of the corners are always, what am I saying? That most of the corners are always carbon what? When you see a corner in organic chemistry, that's a carbon atom. Now, you people, this is ribose. That is ribose. So, and that is the R in RNA. So the D in DNA stands for deoxyribose. Stands for what? Deoxyribose. So you may realize that ribose is still there as a complete word. But then we are having this deoxy part. You people, what do you think deoxy means? When you hear deoxy, I think you people know what D means in science. I told you D means removal. D alone means removal. And so what oxy may mean? Oxy may mean oxygen. So what is deoxy in full? D means removal. Oxy is completed as oxygen. So what do you think deoxyribose means? What do you think deoxyribose means? When they say deoxyribose, they are meaning that remove one of the oxygen atoms from ribose if you really want to come up with deoxyribose. Simple as that. As simple as that. So I'll come back here and tell you now. Are you seeing this carbon number 2? So you may ask yourself, where are we really going to remove the oxygen from? Because we are having this oxygen, we are having this oxygen, we are having the oxygen here, oxygen there, and oxygen there. So you may be confused and be like, where am I really going to remove that oxygen from? So this is what now they call learning by heart. You may ask yourself, how can I really learn this? It has no explanation. You just need to appreciate that the oxygen is going to be removed from carbon number 2. Carbon number 2. That's what you're supposed to know. Carbon number 2. So when we come to carbon number 2 and remove, when that O is removed, ribose becomes deoxyribose. I don't know if people will be in position to recall that. Carbon number 2. Carbon number 2. For me, I used to recall this. I used to say ribose is becoming deoxyribose. So it's only 2 sugars involved, and so we shall involve carbon number 2, period. Ribose is becoming deoxyribose. We are only having an involvement between 2 sugars. There are only 2, ribose and deoxyribose. So because there are 2, let me learn by heart that it's from the second carbon atom. Something like that. So when they say deoxyribose, they are meaning remove oxygen from the second carbon atom, and then your ribose will become deoxyribose. Yes, Fahad. So people are following. Make sure you understand the structure.
My concern is that there is sometime when on that carbon atom 3, the OH is inside. When is that happen? Or what is it exactly?
But are you sure it is always inside in this topic? Or you have looked
Like
at a structure that looks like that, and the OH is projecting inwards.
Like from, is it FA? Yeah.
You are talking of it was under carbohydrates, isn't it?
Yes.
And these are nucleic acids. And they were telling you about the isomers. I think it was the fumarose forms of glucose.
Okay.
That's when we have such projections inwards and outward.
Okay.
But that will
for
come.
this,
For now, first understand ribose, then deoxyribose. That will come.
okay, thank you so much.
Okay, welcome. So don't be like, but I've seen it. No, first understand how ribose looks like, then deoxyribose, so that you'll understand the rest as side information. So I'm now going to condense this structure. Just because your curriculum, they no longer set objectives, but this is an objective we used to enjoy those days. Yeah, they put for you a structure and ask you which of those structures deoxyribose. For you, just look for carbon number 2 and you look for that carbon without oxygen. And then you leave. So let me condense this structure. So you allow me condense it. But before you condense it, you now know how ribose looks like and how deoxyribose looks like. Now, what did I say? What did I say? These two things, the ribose and deoxyribose are pentose sugars, isn't it? Or pentoses. So that means the pentose sugar is the very first component of the nucleotide. Is the very first component of the what? Of the nucleotide. Now, are you seeing this A? This A means acid. You understand? That A means what? Acid. So what gives this nucleic acid the acidic nature? It is phosphoric acid. That means phosphoric acid is another. Phosphoric acid is another what? Is another component of a nucleotide. Are we together? We are using the polynucleotides and how they look like to help us find out what are the components of just a single nucleotide. And we have so far found out the pentose sugar, and now we have found out the phosphoric acid, and you now know it's what gives those things the acidic nature. So the other last component, you're going to just appreciate it. But I don't always want to tell it to students this way. I always tell students that this N will now help us get the last component. But remember, this N in DNA is nucleic. But here I come and tell you that the third component is a nitrogenous base. Is a nitrogenous what? Base. That means before we even look at the whole nucleotide, we need to know what are pentose sugars, which we have looked at. I'm now supposed to teach you nitrogenous bases, then after I'll go to phosphoric acid. So when they ask you for the three components of a nucleotide, pentose sugar, you know how I've derived at it. Phosphoric acid, you know how I've correctly derived at it. Even the pentose sugar, we have correctly derived at it. But the nitrogenous base, we have wrongly derived at it. But it is part. It is part. It is part. So when you're talking of nucleotide structure, components of a nucleotide, never forget the nitrogenous base. Never forget the what? The nitrogenous base. Now, you now know the pentoses and how they look like. As I condense the structures, I'm now going to take you through nitrogenous bases. Let me ask, you people tell me in the chat, do we have people here who have never heard of the word purines and pyrimidines? Let me see those who have never heard of the words purines and pyrimidines. Because I may assume you have content, yet you are hypodatic when you don't have data. I want to see those people who have never heard the word purine and pyrimidines. Because I may never had. Wait, people, what have you heard of? Hey, you've never heard of those things. Hey. Yeah. Okay. So let us first compress these structures of the pentose sugars. Then we talk about the nitrogenous bases. Hey. So people are hypodatic. I thought you people completed the syllabus and you just want us to revise these things. So the pentose sugars, we now know there are 5 carbon units. Now I'm condensing, eh? Yes. So when they ask you for a condensed ribose, you just need to do this, put here the O, do this, you do that. You understand? Like that. So they are only corners. But what am I drawing? Am I drawing a bat? Then you all know that here we always have the carbon, isn't it? Then here we always have the carbon. Hey, sorry. You all know that this is carbon number 1, this is carbon number 2, and this is carbon number 3, this is carbon number 4, and this is carbon number 5, like that. So you know that here on top, that's where we have our OH, but for this way down, it's like that. So basically in biology, what you not only show the carbons if you're condensing it, you're supposed to keep showing those hydrogens like that. Carbon number 2, carbon number 3, like that. Carbon number 4. Come here and show the hydrogen. And the way this carbon number 5 should look is very weird. So I'm going to make it a bit longer so that you see the complete structure. So it's supposed to be CH2OH. If you people recall it, this is carbon number 5. Remember, it had 2 hydrogens and the OH. So now, if you're to draw ribose, this is how you should draw it. That's ribose. Then if you want to come and draw deoxyribose, now you come to this OH and make it H. So you remove the hydrogen. So you remove the oxygen. So when you remove on carbon number 2, you come up with a new structure, which is a structure of deoxyribose. So I'll come and draw it also this side. It will also be like this one, like that. Hope people are copying not slowly and taking note of whatever I don't speak. This. Then you even start with this one and put here H. So on top, inside it has just remained H. Then the rest of the things are just like the way they are. Then this one, CS2OH. This one has remained as H. This one OH, so the rest are intact. It is only that OH that becomes H. And this one is H. And is OH. You see here there is as we are drawing together, but let them give you a paper and pen and tell you to start. That is carbon 1, carbon 2, carbon 3, this is carbon 4, this is carbon 5. So I think you can see that what was OH has become what? H alone. So that is now the deoxyribose. Yeah. So this is the deoxyribose. And I've told you by condensing here, we simply mean don't show the carbons, just use corners. So you people now know these two. And please keep them well. Keep them well because now the real biology is coming. We are now just joking because we are just drawing covalent bonds. We are going to start condensation and you will see that things are not actually very easy. as you may think. Yeah. Okay, now I'm going to take you through the nitrogenous bases, because most of you were like, you're hearing them for the first time and so on and so forth, which I didn't know. I thought you people actually know something. So you're like, we remove O from carbon number 2. Yeah. So in biology, 2C means 2 carbons and C2 means carbon number 2. Yeah? Hope you get that. Now we are going to go to the nitrogenous bases. Most of you have said you've never had. You people, what do you know about nitrogenous? You people, when they say something is nitrogenous, you know what we call the nitrogenous wastes. Do you know the nitrogenous wastes? What do you know about nitrogenous wastes when they say something is nitrogenous? Because now you need to understand that. So 2, we are now going to talk about the nitrogenous bases. No one is giving me an idea? Okay, they have nitrogen, but no one is giving me my answer. Yeah, there's a certain answer I wanted. Let me write to save time. Eh? So someone is like, they contain nitrogen, which is right. But what I preferred is that they contain. Yes, Alan. Let me hope you have the right answer. What do I want?
Nitrogenous base, it is divided into 2.
Good night. They contain nitrogen, or I can say they contain amino groups. I don't know, but I remember when I was teaching you, amines, I remember telling you, of course, nitrogen is just N, like this, is just N. Even the amino groups have N, yeah? But this is the most important thing, that nitrogenous bases should have the amino groups. They're the ones that make them nitrogenous, eh? And you know that amino groups, I remember telling you, they can be like this, with only one group attached to them, or with only one hydrogen of ammonia replaced. They can be like this, when 2 hydrogens of the ammonia have been replaced, and they can be like this, when all the hydrogens of the ammonia have been replaced. That's why we had things like methylamine. That's why we had things like dimethylamine. That's why we had things like trimethylamine. So you take note of that. Those are the nitrogenous bases. So they have this. And these ones are the best 2. Yeah. Yeah. Why this one may not be so good, it's because it's having no hydrogen, so it may not help us to do what we want. Please leave our screen. Now, someone may ask themselves, what do you mean? What are they? What are these nitrogenous bases? What are they? Okay. Who was that boy? Alan, raise up your hand again. Yes, now you can tell me your stories. You tell me your stories, but be very fast. I'm giving you half a minute. Tell me everything so that your friends get motivated.
I was saying that we have purines and pyrimidines. Then under purines, we have guanine and adenine. Then under pyrimidines, we have cytosine, thymine, and uracil. Yeah,
Tell me about
that's
their
it.
structures. Tell me about their structures also. Purines are what?
In fact, if we want to make DNA, we have those others, but apart from uracil. Then RNA, we have those ones apart from thymine. Yeah, that's it.
Okay, that is good. That is good. That is good. That is good. I just wanted your friends to get motivated. So I think you're hearing you don't have content alone. When you be there and you feel hypodatic, just understand that that is happening to only you in the whole Uganda. People are reading and people have content. People are hungry for medicine and surgery. Yes. Okay. Now, I'm going to tell you something. I'm going to tell you something. I'm going to tell you, I'm going to tell you a story. If you're tired of my stories, you log off. Yes. I got maximum marks in biology. Maximum marks in biology. Okay. The story is this. If I show you something like this, A, B, C, D, E, and F. These are what they call letters of the alphabet. These are what they call letters. Okay, they are called letters, and what do they make up? They make up the alphabet. Or we can call it the alphabetic code. Isn't it? It goes up to Z. I want you to listen. Hey. Now, the nitrogenous bases, for them, they make up what they call the genetic code. So I'm going to first list them here, so you understand in terms of back titration. I'm going to first list all of them. At your level. At your level, nitrogenous bases are 6. Are 6. They are 6. Nitrogenous bases are what? Are 6. So we are going to first list them. Remember, you don't know them, so you listen. The first one is guanine. You can have your own order. Guanine. The next one is adenine. The next one is cytosine. We have thymine. We have uracil. There is a way uracil refuses to look like the friends, and we are also going to see how it misbehaves. So, actually, the other 6th one is very rare. It's only in Michael Kent. Let's take 5. Yeah, consider them at your level as only and only 5 that we study. So those are the ones. Now, guanine is represented by letter G. Adenine by letter A. Cytosine by letter C. Thymine by letter T. Uracil by letter U. You get? So these ones I call them the 1-letter codes. Yeah. 1-letter codes for those bases. 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 1-le I want you to follow. So instead of guanine, I'll be just showing you G. Instead of adenine, A, cytosine, C, thymine, T. I think you can see where we are getting them from. G for guanine, A for adenine, C for cytosine, T for uracil, T for thymine, U for uracil, C for cytosine. Yeah. So those are the one-letter codes. So just like the ABCD up to Z gives us the alphabetical code. But remember, I've told you that these ones are just what? Letters. If at all we go to our guanine, are you listening? Adenine, cytosine, thymine, uracil. These ones are not letters. I've told you they are the nitrogenous what? Bases. And these ones don't give us the alphabetical code. These ones give us what they call the genetic code. So at least leave this class when you know that the genetic code is made up of nitrogenous bases, unlike the alphabetic code that is made up of just weird letters. And these ones are just ABC. Leave alone those things you used to do in primary. A, axe, B, banana, C, cockroach, D, what. This is science. This is medicine. Guanine, A, adenine, C, cytosine, T, thymine, U, uracil. Yes. Okay, now you know what they are. Now we are going to divide them. We are going to do what? To derive. So we are going to divide them. How are they divided? So all of you people who are giving me your things of small purines, pyrimidines, you're very bad. You cannot just tell a person out of the blue those things. So how are they divided? You now know that there are 5. So nitrogenous bases. How are they divided? So I want the Baganda to listen carefully here. Nitrogenous bases are divided into purines and pyrimidines. Pyrimidines. Divided into what? Purines and pyrimidines. Yes, very, very nice words, sounding very well. Purines, pyrimidines. So now we need to know, out of the 5, which ones are the purines, which ones are the pyrimidines. Get the point? Now, in Buganda, there is what they call abusive language. Yes, there is what they call abusive language. And I want a Muganda who knows Luganda very well and you know how to abuse. Raise up your hand. Raise up your hand. Trevor. Trevor is going to help me. Yeah, at least I trust him. So Trevor, I want you to tell me. When you annoy an African mother who is a Muganda, I'm talking to those ones strictly. And then they annoy you as a family. Maybe they find you with your siblings very disorganized. Then they tell you, then the mother says, Galabe. I'm not going to add on another word. But then they tell you Galabe. What do they mean? Okay, let me make something. Let me make it more clear for people to understand. In Uganda, if you're not a Muganda, you're going to understand by force. Because you're going to forget these examples and you're going to disturb us. In Uganda, I repeat, there are 2 kinds of abusive languages. There is a way they abuse big people, and there is a way they abuse people who are small. There is when they use ka, then there is when they use ga. Which one do they use for small people? And use gana mwe, kana gwe, karabe, gulabe, galabe, lulabe, lirabe, gulabe. Who can tell us? No one is going to tell us. Yes, Fad. Other people are shy. Ama, you tell us.
Like a galabe abat is meaning big ones.
Big ones also. Galabe, they are meaning big ones
Yes.
also.
Yes.
That's the only thing I wanted. So if you've not had now, you wait for exact biology. But that's what I wanted. So in Buganda, have it in mind when they say galabe, when they use ziga, they mean something very big. When they use ziga, they mean something very big. So I want to, before I even proceed with what I want, I'm going to show you two structures. I'm going to show you two structures. We have what we call the hexagon and the pentagon. So that's the hexagon. That's the hexagon. Then we have what we call the pentagon, which is like this. Now I'm going to make that pentagon, this one, I'm going to make it touch this one so that it's like this. Like that. You know that people are getting. I drew two hexagons, but I made one pentagon touch this first hexagon. That's what I have. Now, when you look at those two structures, this one is bigger. This one is bigger, but this one is a smaller structure. Why is this one smaller? Because both of them have the hexagon, but this one has an extra pentagon. So it's a bigger structure. I want you to listen. I want you to listen. So after understanding that, those are the actual things actually. So this is the real science. Hexagonal ring and the pentagonal ring, then only the hexagonal ring. First get that. Then we go back. So what you always do for these purines, you always come and abuse them. The way we abuse them in Uganda. Then you say, gapurine, like that. Gapurines. Nze gapurines gantama. Nzagagana gantama. Like that. You forgive me for the language barrier and so on and so forth. But if at all that is very hard for you, you just understand that the purines are two, guanine and adenine. And because Ga means very big, they are taking on this structure. That's what I wanted. This thing is hard because you're supposed to understand how their structure looks like and the examples. So I've told you, how do we know that the structure is bigger or smaller? Have it in mind, we have hexagonal rings in both cases, hexagonal. But one of the structures has an additional pentagonal ring, which is just like that. You understand that and proceed. Then you go back, purines and pyrimidines. What am I really going to do? To recall the structure and examples, you come and abuse purines. Be like gapurine, gantamans. Gapurine. What do you mean by gapurine, guanine, and adenine? That they are purines. And the mere fact that you are abusing them using ga, that means they are very big. So they are taking on the bigger structure, which is that of the hexagonal ring plus the pentagonal ring. Then the rest of the bogus is the other side of the pyrimidines. What is the bogus? The cytosine, the thymine, and the uracil are pyrimidines. And what is their structure? A single hexagonal ring. As simple as that. Biology has a lot of content you can understand, but memorizing is always a bit hard. So as you read, I told you that I'll get a session next year to tell you how you can memorize some of the things, but that comes after you've mastered the content so that you're just correcting errors and eliminating errors. So that is it. That is it. I don't know if you need me to pardon, but someone can maybe pardon in the chat. But that's all I can say. Yeah. That's all I can what? I can say. Now, I meant that purines, the first one is what? Guanine. Then the other one is what? Adenine. Guanine is G, adenine is A. And then we are saying that their structure is what? Bigger, because of the reason you know. And what makes you say that the structure is bigger? They have the hexagonal ring. They have the hexagonal ring and the pentagonal ring. Pentagonal what? Pentagonal ring. Then you know that this one is just the hexagonal ring for the pyrimidines. So what is the balance after putting off the guanine and adenine? The balance is what? Cytosine. Thymine, not thiamine. Thiamine is a vitamin. Eh? Thymine, not thiamine. Then uracil. You can see uracil doesn't resemble the friends. Doesn't resemble the friends, and we shall see how it misbehaves. I don't know when. So, I now want questions. I now want questions on just the nitrogenous bases, because I want to leave them and now teach you phosphoric acid. That is chemistry. And that's what I like most. So I now want to go to phosphoric acid. Any questions as far as the nitrogenous bases are concerned? You people, should we proceed? If you're saying that we should proceed to phosphoric acid, you type in the chat phosphoric acid, phosphoric acid. Because most of you don't know how it looks like and you need it. Most of you don't know how it looks like and you need it. Remember, it's now the third component of a nucleotide. Remember, there are three: pentose sugars, then nitrogenous bases, which we have discussed, and what they have, how they look like, how they come up with the genetic code, and so on. Then three, I want us to look at phosphoric acid. Yeah, they will give you a very hard question, which is a scenario, and you see the word purine, thinking you can attempt. Then besides the question, you put there gapurine, but you won't even reason the question. And you'd end up just putting that for the examiner, and you will give up. So I think no, they never ask you for the structure of those things, so don't mind about them. So phosphoric acid, all of you know the molecular one, but for us, we want the structure. I will explain to you the details when you reach that topic of drawing structures in chemistry. But when you're drawing such structures, you're supposed to first appreciate who the central atom or what the central atom is. And when I look here, these atoms are many. There are 3 hydrogens and 4 oxygens. So hydrogen cannot be the central atom because it's more than 1. Oxygen cannot be the central atom because it's more than 1. But I have a chance that P is alone. So P will be there. So I'll put here P. Yeah, it is the central one. Now, in chemistry, I'm just saying this and I'm not writing it. The number of hydrogens, provided the compound is having both oxygen and hydrogen, the number of hydrogens represent the number of hydroxide groups or the hydroxyl group attached to the central atom. Listen carefully. Provided you're drawing a structure of any compound that is having both hydrogen and oxygen, the number of hydrogen atoms represents the number of OH groups attached to the central atom. So because the hydrogens are 3, that is clear that the number of OH groups attached to the P are 3, because even hydrogens are 3. So you just come and do that. You come and put here OH. Come and put here OH. You come and put here OH. Then after that, you realize that hydrogens are done. You have used only 3 oxygens and they are 4. You get? So, and the oxygen alone is always okay with 2 bonds. So it is left with no option other than doubly bonding to the central atom like that. And you all know phosphorus is very okay with the 5 bonds because it's atomic number 15 with the electronic configuration 22825. So that means it is very okay with what? With those bonds, 5 of them. As a student who doesn't do chemistry but you do biology, you have to struggle a bit. But of course, you just need to put in more energy. So this is how phosphoric acid looks like. This is the structure. Structure, or you can call it structure formula. And you need it. You need it. You need it. So after you people understanding that, I want someone who can define for me condensation. So you people, hope you have gotten the structure of phosphoric acid. The other one is molecular, the one you knew. This is the what? The structure. I want you to understand why there are 3 OH groups, because the hydrogens are 3 in a compound having both what? Hydrogen and oxygen. Why is it like this? Because we had been left with only and only one what? One oxygen, yet is okay with 2 bonds. So it was left with no option other than doubly bonding to the central atom. So that's not left out. And by default or by chance, the phosphorus came out to have a total of 5 bonds, which are the exact bonds it needs because it's in group 5. Get it? Like that. So now, who can define for me condensation reaction? Condensation reaction. Yes, Fahad. But Fahad, your background, I don't know what's wrong with your background. Yes, blessed Akrod.
Yes, doctor. The condensation reaction is the one that involves loss of water molecule.
Is it only water?
For me, what I know is that a condensation reaction, it can also mean a dehydration reaction. Okay, a reaction involving loss of water molecule.
Oh, that is good. That is very good. So, condensation reaction involves loss of small molecules. Why I want to give you this definition early enough because we shall find it in polymerization. Yeah? And of course, we are talking of formation of polymers, polynucleotide, polypeptide. So we are losing small what? Molecules. The small molecule can be water. You will see when the small molecule is HCl. You will see cases when the small molecule is ammonia and etc. But water is the usual one. Small molecules usually what? Usually water, but not always water. This can be lost, this can be lost, provided the molecule is small. That makes it a perfect condensation reaction. That's what I may say. So you people, we are going now to talk about condensation. We now know the components of a nucleotide and how they look like. So let's ask ourselves, how do they interact to make the nucleotide complete? You get it? That's what we are going to look at now. So we said the nucleotide is having two components, sorry, three components. The very first one, we said pentose sugar. Pentose sugar, of which we talked about it very well. The next one, we said the nitrogenous base. Then the other one was the phosphoric acid. Yes, isn't it? Now, our next thing that we are going to do, you know how all of them look like. So we are asking ourselves, how do they interact? How do they interact? How do they interact to? How do they interact to form the nucleotide? Yeah. So all you need to know that here the pentose sugar is the main guy. Yeah. In whatever we are going to be doing, the pentose sugar is the main what? Is the main guy. As we are coming up with the structure, just make sure you respect our pentose sugar. It's the one that is going to lead all interactions that are coming in. And without it, we cannot have a complete nucleotide. So you listen carefully. You listen how? Carefully. So, you look here. Formation of a what? Formation of a nucleotide. Formation of a nucleotide. So I've said that first things first, draw our pentose sugar. So I'm going to come and draw it like this. Okay. Isn't it like that? So you all know that first things first, here we are having the CH2, and we all know this CH2 has the OH attached, like that. Then we all know that this carbon is having OH on top, and we also know it's having H down, like that. I'm not going to show other carbons for a reason. So this is carbon 1, this is carbon 2, this is carbon 3, this is carbon, this is carbon 5. I think you know that. So I've told you that I'm not going to show other carbons for a reason. So you know that this is our pentose sugar, and I've already warned you that it is our main guy. That's why I'm drawing it first. Follow critically, because as you're revising, you'll have to do these things on your own. Then later you confirm whether what you're doing is what is right. So please and please, only and only concentrate. Only and only concentrate on these carbons. Someone may be like, why have you only talked about, or why have you only exposed stuff for carbon 1 and carbon 5? I have a reason as to why. But, okay. Let me expose this also. Remember, this one has OH down like that. So someone may ask, why have you really exposed? Why haven't you talked about this? Carbon 4. Because you all know it has no OH. It's only having hydrogen, which hydrogen is everywhere. So it doesn't have that OH to make it special. Someone may ask, why haven't you exposed this? Because that too has issues. It is from that carbon number 2 that we remove O to convert ribose into deoxyribose. To confirm that you people are actually listening to me, who can raise up their hand and tell me the reasons I've given as to why I've not exposed all the bonds at 4 and all the bonds at carbon number 2. Because that's how you will cost us. It'll be like I never taught, yet I said. So who can quote the exact statements I've made? I think it's only Steffi who is listening at me. The rest of you are not listening. You are very bad. What have I said, Wamma?
You said at carbon number 4, the carbon
Mhm.
only has a hydrogen atom attached to it, so it does not have like a hydroxyl group that's making it special from the rest. Then
Exactly.
at carbon number 2, that's the carbon we always consider to convert ribose to deoxyribose by removing the oxygen. Thank
You
you.
are listening. Thank you for that. Thank you for that. So that's what I've said, people. That's what I've said. So if you're not listening, you are doing that alone. Yeah, you're doing that alone. People are listening. Now, after having the pentose sugar, we should ask ourselves. So, how do they interact? I'm going to answer this question. How do they interact to form the nucleotide? The answer is easy. It is by condensation. Yeah. So it is through condensation reactions that those components interact to give us the nucleotide. So that means if we are having the pentose sugar as our main guy, which we already have, it is supposed to condense with the nitrogenous base. Are you listening? And it's supposed to also condense with the phosphoric acid. But the question is, where, at which carbon number is this phosphoric acid going to attach, and where this nitrogenous base is going to what? To attach. Simple as that. That's what you need to know. That's what you need to know. So we are having 3 carbons. 3 carbons that we have exposed. We have exposed carbon 1, carbon 3, and carbon 5. Let me begin with the phosphoric acid. Because this phosphoric acid, I'm telling you how you can memorize these things because they can confuse. Trust me, they can confuse. By the time you bring in coordination, this content goes away. So you must have a few things to help you recall some stuff. So for phosphoric acid, because this phosphorus is in group 5, because the phosphorus is in group 5, the condensation with phosphoric acid will occur at carbon number 5. Hope you're listening. Hope you're listening. Hey, because that phosphoric acid, but that is a local reasoning. The books will just tell you carbon number 5. They will not explain why. But you should come up with your own way that you can easily memorize something small and you proceed with the paper. Just not remembering that carbon atom can bother you so much and be like, can I really do this number? Was it carbon 3? Was it carbon 1? Was it carbon 5? So you should recall some things. I think we should put it here as a note. Phosphorus. But I don't want you to focus so much on these things. These are wrong things, but enabling us to recall, yeah? Phosphorus is in group 5. Hence, condensation with phosphoric acid also. Oh, people are writing with phosphoric acid. Panai will occur at carbon number 5. So do you see 5 means carbon number 5? 5 means 5 carbons. You get the point? So I'll come here and draw my phosphoric acid. Hope you people now it looks like. It is easy when I'm drawing them. You get a book and a pen. You find yourself scrolling on TikTok. Like that. You get? Now the question is, so the phosphoric acid is done. Pentose sugar main guy, it should condense with others. It has condensed with phosphoric acid at position number 5 for the fake reason I've given. Now the question is, what of the nitrogenous base? What of the nitrogenous base? So we now have carbon number 3 and we also have carbon number 1. So should be like, which one really is going to take the nitrogenous base? Because we are also supposed to have what? Condensation here. I don't know how you're going to recall this. I don't know how you're going to recall this. But you people, if you're to, okay. You look at that structure of DNA because you recall these YouTube videos, I'm sure. I want you to look at those structures of DNA. Are you seeing them? I want you to look at those structures of DNA because I think you've seen them before. I want you to just look at them just like the way they are there. I don't know whether you people realize. Okay, that is good. I don't know whether you people can realize that this is how the things have been. Where are we? Are we here? Okay. I don't know whether you people have realized that the DNA, there is a way it's having strands, and then the bases are coming off like this. But they are coming off at an angle of 90. I don't know if you people have seen that. Maybe people have you seen that? That even if it's like this, that these things, the lines that are coming out like that. Don't you see that they are coming at an angle of 90? And I know you've seen the names which have been there. They have been putting guanine, adenine, thymine, cytosine, the things they have been putting. So, what I want you to understand from that nitrogenous bases always project at an angle of 90. They always project at an angle of 90. I don't know. Maybe you can look at the angle of 90 again. But where am I going to get it from? How come I've lost that screen? What happened? I had that screen. What happened to that screen? It's bad. Did you people see did you see the 90 stuff, the 90 degrees? Yes? Did you see the 90-degree stuff? Because now you're supposed to learn, you're supposed to not only learn the content, but also you're just disorganized. You see, some people didn't see. That's what I hate. You look at the screen. You look at the screen. You look at the screen. Are you seeing that video? We are going to watch these videos. You're going to watch these videos. Are you seeing how the DNA looks like? Are you seeing the G, the A, the A, the G, the T, the C, the A? Do you see them coming off at an angle of 90? Do you see them coming off at an angle of 90? Right angles. Do you see them coming off at an angle of 90? Even here. I don't know if people see the angle of 90 throughout. Angle of 90, angle of 90. Even here, do you see this one? Angle of 90, angle of 90. And we are going to watch all these ones. You see? Angle of 90 throughout. I think we are together now. Not so? Okay. So you people, if these nitrogenous bases are coming off at an angle of 90, where is the nitrogenous base going to attach? Carbon 1, carbon 3. Carbon 1, carbon 3. If the nitrogenous bases are projecting off at an angle of 90, which one is the carbon that can hold the nitrogenous base at an angle of 90? Is it carbon 1 or carbon 3? Because carbon 5 has already been used, so it can't be back. That is good. Carbon 1, that's what I wanted. So it's supposed to be carbon 1. So our nitrogenous base, we have said it will come there. But remember what I told you, that this nitrogenous base, it can have something like this. The N can have the H, you get? Then this one. But I also told you, don't be surprised when it can have maybe the H alone like that. Maybe this is a double bond, something like that. Remember, N must have a maximum of 3. And I told you the groups which are better are those ones with hydrogen. Because maybe without hydrogen, we cannot have what we want. So you people, that is it. Phosphoric acid, nitrogenous base, carbon 1. And why have we chosen carbon 1? Carbon 1 because the nitrogenous bases must project at an angle of 90. Because the nitrogenous bases must project at an angle of 90 degrees, like that. Yeah, must project at an angle of what? 90 degrees. But remember we said these things should interact via condensation reactions. Where condensation reactions, we are losing small molecules. You get? And the small molecule, it is usually water. And we are going to lose only water for biology. The other things I told you, they're in polymerization, which is chemistry. So for biology, I want you to only think about water. Hey, don't overthink. Eh? So by usually, I meant in biology, but when you reach chemistry, even others. So as a chemistry teacher, I cannot say loss of water. Though in biology, we just say loss of what? Water. Okay, now I will come and ask myself, how are we really going to lose the water? It's not obvious. Remember, the phosphoric acid is having the OH, not so? Even the pentose sugar is having the OH, but remember, we just need water. Yes, we just need water only. So that means water can be formed. How? The acid gives over its OH because for it, it's having so many OHs. So it will give over its OH, the whole of it. Where are we? How can I show this OH? Let me change the way this OH is formed. Let me just extend it a bit. This is the OH, like that. Then let me also write this OH for the pentose sugar. Let me put H here. Why I'm putting H here? Because I want us to lose the water well. Remember, water means one OH and one H. That's why I had to put the OH near the H. So don't overthink. Don't say that, but you've changed the structure of not. Okay, now I'll come and say, since the acid has so many OH, it will give up all its OH. Then the sugar will give over only one H, and then we shall lose the water molecule like that. So that's what condensation means. You understand? Then you also come here at carbon number 1 and be like, we need to lose water. But of course, this time round, we have no option other than losing the whole of this OH and then maybe one of the hydrogens from the nitrogenous base. But the nitrogenous base does not have O. So that means the pentose sugar should give over the whole of its what? OH, so that we can lose another water molecule. So this is what they mean by condensation. So yeah, condensation has happened. Condensation happens. Remember, you're going to convert this thing into statements. This is now the structural biology. You need to make statements out of this very well. So people have noticed the positions where the condensation has happened. Now, hope you have seen how the three interact to form a nucleotide. So now the nucleotide is what? Is complete. And how is it looking like? So I'm going to come and draw the complete structure. The complete structure. The complete structure. Complete structure. So I'll come, I'll draw. See that? Then we all know that this is where we shall have our nitrogenous base. You get? Yeah, I'm just trying to show that in because it participated in everything. Then you all know that we are having our OH here remaining. Maybe it's waiting for something that we are about to see. This is our pentose sugar. Then you very well know that this other one interacted with the acid. It was like that. Then we remained with, I think, P here. We had O there. Then here we had a P. Then this side had the O. Then here we had OH. Then here we had OH. So that's what they call the complete structure of a nucleotide. That is it. I don't even want to add on other things. But if you want to really name, this is carbon 1, this is carbon 2, this is carbon 3, this is carbon 4, and this is carbon 5. Numbering is important. I told you we follow the clockwise direction. So that is one of the things I wanted us to cover today. Because now after we have this, other things become very, very easy. Because this is now the nucleotide. Now forming the polynucleotide is easy. Just like the way you guys had the amino acid and getting the polypeptide was easy. So we now have the nucleotide, and now getting the polynucleotide is going to just be very obvious. After we are going to go and read the statements, and then I will ask someone to repeat the same statements. but nothing much. They will not over deviate from this. Yes, it will be like the pentose sugar condenses with the nitrogenous base at carbon number 1. Phosphoric acid condenses with the pentose sugar at carbon number 5. And that's how we form the what? The nucleotide. You can talk about loss of water molecule even after you mention the condensation. In biology, it's very okay. Like that. So our number 3 is left intact. For a reason. So, are people ready? We now look at how the polynucleotide comes about. Are people ready? We talk about how the polynucleotide comes about. Or when you tell me that you're ready for it, I will assume that everything has been okay. I will assume that everything has been okay. Someone is like, where are those hydrogens and so on? Those things are still there. I think I told you, but you all know that what is remaining right now is the H. You know what we have lost. Carbon number 1 is remaining with only H. Carbon number 2 has its things, but we just don't involve it in these things for a reason I gave. Carbon number 3 has its things, all of them. We have not tampered with it. Even carbon number 4 is still having its hydrogen, not tampered with. Carbon number 5 has its 2 hydrogens. We have only interrupted its other things. That's what you should understand, only that. Okay, now I have a question. I don't know whether I have enough chemistry students here, but I taught you what they call chemical equilibria. And in chemical equilibria, I told you what they call esterification. I told you what they call what? Esterification. Who recalls the things that must react to form an ester? Who recalls the things that must react to form an ester? Who recalls the things that must react to form an ester? Yes, Akron.
Yeah, to form an ester, we react an alcohol and a carboxylic acid.
Exactly. That is enough. That's what I wanted. But as you grow up, you will realize that it is strictly a reaction between an acid and an alcohol. Provided those two react, whatever you get will be an ester. Ester formed. And because we have the ester formed, we can also say, hence, ester bond is also formed. Shall I say that ester bond is also what? Ester bond is also formed. So every time you react, you understand, every time you react an acid and an alcohol, you get the ester. So if you're like, what is this ester and so on, I'm referring you to chemical equilibria. So because of that, we can also say the ester bond has been what? Has been formed. So when you look at this condensation that happened, this condensation was between, remember, the alcohol is something that is having the OH group, nothing much. So don't think alcohols as what? Don't think of alcohols as other things. Maybe ethanol, methanol, and other stuffs. Provided something has the OH group, it is very okay, and we are very right to call it an alcohol. So, just a moment. Just a moment. So I've been telling you that in case any acid reacts with an alcohol, by an alcohol means something that has the OH group present. I'm teaching a marking point. What we form is what they call an ester. So an ester is formed. And since we say that an ester is formed, we are right to say that ester bond is formed. We are right to say that the ester bond is what? The ester bond is formed. We are very right. Now, when you look at what we have just tried to do, this one, look here. This one was phosphoric acid, so it was an acid. You get? Then this pentose sugar provided the OH. So because it provided the OH, it behaved like an alcohol. Because it provided the OH, you understand? This phosphoric acid was our acid. Of course, it is an acid. Then this pentose sugar provided the OH. So that means it acted like an alcohol. So it's like what we formed. Chances are high that it is an ester in one way or the other. So that means we have the ester bond there. So when I call it the ester bond, I don't want you to be so surprised. But because of this P, because of this P, because of that P, I'll be happy to tell you that we have a bond existing there. The one they call the phosphor-ester bond. Yes, the phosphor-ester bond. So, have it in mind that the phosphor-ester bond somewhere there. Yeah. Phosphor-ester bond. So you may ask yourself, why phosphor? Because we are having phosphorus coming in play. Where did the ester bond come from? Ester bonds are formed when esters are formed. When an ester is formed, when an acid interacts with an alcohol. What do we call an alcohol in biology? Anything that has the OH group active. And any acid usually has the name acid on it. So the ester bond was there. But because of the neighboring phosphorus atom, we call it the phosphor-ester bond. Then this bond was just between carbon and what? And nitrogen, both of which are nonmetals. So that bond is covalent. So that's a covalent what? Covalent bond. Hope you understand. Yes, Akrod?
Excuse me, doctor.
Yes, please.
I'm confused.
Mm.
You are calling the Pentose sugar, you're calling it an alcohol because of the OH.
That is active and has participated, yes.
But even the phosphoric acid is having OH.
But it is an acid in this case. But the pentose sugar
So
is
what
not
makes
an acid, but it's only having the OH as the active group.
teacher, what makes the phosphoric acid an acid?
What have you asked?
What makes that phosphoric acid an acid?
Because of the protons. You're saying it is already an acid, so it is an acid. But the pentose sugar, you're not sure which homologous series it is belonging to. But because it has been active in form of the OH, we call it an alcohol. But you've even, it's like asking why is a boy a man? You are asking why is phosphoric acid an acid? You've heard it is phosphoric acid. What else do you want? But the pentose sugar is just a sugar with no functional group. But because the OH is active, it qualifies to be an alcohol.
Okay, I get.
If the phosphoric acid just had the word phosphoric alone without acid there, you may have a point. But it is an acid, so what do you expect? Okay, now that is the name of the bond. So make sure you know those bonds very well. They are very important. Then the other one is just a covalent bond. Now, the question is, how are we going to form now the polynucleotides? We now have the nucleotide, but now we need to form the polynucleotide. I don't know if you people are realizing there are some free things that are remaining on our, you look at our structure. Who can tell me the free things that we have remained with? Do you see any free groups? Do you see any groups that have been left free? Because that's where the point is. Do you see the things that have been left free? You just look at that structure. Who can raise up their hand and you tell me the free things, okay, the groups that have been left free and you think they may be of hope? Yes, Emma. What do you see?
Well,
Tell me what you see from the structure.
yeah, I think the OH on the third carbon is free, and the
Mm-hmm.
OH on the phosphate atom is free.
On the what?
Yes, on the phosphate atom.
Oh, phosphorus.
Most probably. Yeah, phosphorus atom. Yeah, right.
Yeah, that is it. Now, Emma is going to understand how a polynucleotide chain is formed, because that is the only technique. That is the only technique and nothing else. That's the only technique. That is the only technique. Where are we? So this is what our friend has said. This one has been left free. Others have been used free. And then I also realized that this one has been also left free. Even this one is free. And that one is free. Yeah. So for you to come up with that, if you people recall how a polynucleotide was formed. Sorry, a polypeptide. This was the amino acid, not so? People may be like, why are you prioritizing the free stuff? When you brought your second thing like this, I don't know if you recall what you said. This one was giving us the polypeptide. Sorry, the peptide, the first dipeptide after forming the peptide bond. Isn't it? And then, how did you conclude? You were like, we have left a free carboxyl group here, and we have also left a free amino group this side. And we are like, these groups are the ones to undergo further condensation to form a longer chain of peptides, or a longer chain of amino acids. Isn't it what you guys were doing in proteins? Amino acid. How do you condense? This one provides OH, this one only H. You lose the water. You form the peptide link of the peptide bond. Then after you be like, we've left a free amino group here and a free carboxyl group there. So these ones are the ones that will undergo further condensation with other amino acids to form a longer chain of amino acids, what we call the polypeptide. So the same thing is going to happen here. We have left two free OH groups, and those are the groups that are going to assist us as we form the polynucleotide. But before we go for the polynucleotide, it's better you show us the dinucleotide and maybe go to the polynucleotide afterwards. You understand? So I'm going to show people how you can come up with a dinucleotide, because when we draw the dinucleotide, then the poly will be very easy. So look here. So I'm going to do this. So this OH is the only one that is free, as we have said. Then here we shall have our nitrogenous base. That's what I mean. Then this side, I will have my CH2. So this one I'll keep drawing it because I need it, but I don't need the nitrogenous base. So that is our nucleotide, you're all aware. So if at all we want to form a dinucleotide, that means we are supposed to bring two nucleotides together. So let me first bring the other one. Okay. Evidence, we shall draw the screenshots, don't worry. So we shall come and have another nucleotide like this. Then we shall have the OH here, isn't it? Then of course we shall have our nitrogenous base again there, isn't it? Then I will have the CH2. Then I'll come and show, and none of you told me that my structure has a mistake. Let me check the chart. I've just realized it now. Did anyone tell me the structure has a mistake? No one. That's very bad. So we have O here, we have our P here, then we must have OH here, then should have this here, then this one here. So you all know that those are now two nucleotides. You now know what they mean, how they come about, and so on. This is our nitrogenous base. I think we are together there. So I'll just put for you a note here. And put it here. And then we say note. A free OH group of phosphoric acid, condenses with a free OH group on carbon number 3 to form longer chains of nucleotides. I think that is very clear. Yeah, there are just a few things I want to make clear here. So you people realize that our structure had two free groups on the phosphoric acid and then one free OH group on the carbon number 3. So for you to come up with longer chains of nucleotides, you get a free group on phosphoric acid, one of them. Then you condense it with the free OH group on carbon number 3. Then you form longer and longer chains. No rocket science, that's what you do. So I'll come. I have my 2 nucleotides there. Are we together? So I think you can see that this one is now the free OH group on carbon number 3, and this is a free OH group on phosphoric acid. So they are supposed to do what? To condense. And you know, the acid provides OH, the other one only provides H, because the acid has another OH, so it doesn't care. So we lose water. And you know, that's what they call condensation. And because now the other thing has provided OH, it's acting. Hey, it's acting. This acid this is an acid. You get? Hey, let me even put it here. Let me put it below. Maybe people want to disturb me. So this water loss, is what we call condensation. You first note that. After noting that, appreciate that the whole of that thing above, it has acted like an alcohol, because it's only the OH that has participated. Because it is just a nucleotide. So like it's having so many functional groups, but only the OH has acted. So you assume it is acting as an alcohol. Then for this downer nucleotide, the phosphoric acid is the one that has contributed, or it's the one that has participated in the condensation. But remember, it is an acid. So it is acting like an acid. It is acting like what? An acid. I want you to follow. Hey. So because of that, another ester bond is formed. Another what? Another ester bond. Another ester bond. Another ester bond is formed. You are not going to cram anything here. The moment you cram, that is a choice. So another ester bond has been formed for reasons I've given you. So now let us draw the new structure. Let us draw the what? The new structure. So the new structure, the other one is free. So I think people can also realize. Oh, let me first draw the new structure. Let us first see the new structure that is coming out. Hope you know that these things can be reversed. Yeah. Condensation. After condensation, you can have hydrolysis and we go back. So this is the new structure. This is the new structure. So we are having there. Like that. CH2. Then this side you're having the O. You're having the P. You're having this one. Having the OH like that, isn't it? Then here you're having the nitrogenous base. Then here, what was OH? We only remained with O, and then we now touched the P. So the P remained with its double bond with oxygen there. What else? Our P remained with its OH here. But remember here, it was initially touching our CH2 of another nucleotide, carbon 5. Hey, carbon what? Carbon 5. So that means I'm going to come here. This one is supposed to be like this. Like that. You see that? You all know that here there is our OH. Then you know that here we are having our what? So in your free time, you're supposed to sit down and see if you can do these things again and label them very well, and all your bonds are there. You get? The other bonds, the H, the what that are supposed to be there, I know you know. But in case you are in an exam, undo them the way I'm doing them. You're very safe. You're very safe. You're very safe. They shouldn't even lie to you. You're very safe, because after you're going to convert these ones into statements, which statements are most important. You get it? So this is now the new structure. If you people can see. If you people can see that. Now, this is the problem. This is the problem. Now, what do we have? We are having this. We are having our ester bond being formed here. Remember, this is phosphor. But we have this ester bond which was there before, isn't it? Then we have this ester bond that has just come. You get the point? We had the phosphor there already. Or we had the phosphor-ester bond, if you people recall. But now again, we have had the acid-alcohol interaction. So every time condensation happens, this is what I wanted to put in this space, that every time we have condensation happening, we have acid alcohol interaction. And every time we have acid alcohol interaction, ester bond is formed. So we had it at first, and we had the phosphor-ester bond. Now we have had another condensation. So that means we have formed another ester bond. So have it in mind that the whole bond that is there, the whole bond there, towards our wholesomely, that bond is what they call the phosphodiester bond. So the phosphor is still there, one phosphorus, but now the ester bonds are two. So it is now diester bond. Let me tell you, what makes you a perfect biology student of inheritance or the structure of these nucleic acids, when you can explain to someone why it's diester bond and the first one is phosphor-ester bond. If you can do that, then you're just perfect. But if you can't do it, then you please make research and make sure things make sense. This is phosphodiester. The first one was phosphor-ester because condensation had happened for the first time. But as we go into the dinucleotide, condensation comes in for the second time. You understand? And because it comes in for the second time, the thing is now diester. You get? So here we have been talking of formation of dinucleotide first. And I think we are done. Yeah. So what we have here is what they call the dinucleotide. Yes, what we have here is what they call the dinucleotide. The dinucleotide. Yeah, that is it. But if you also realize, the dinucleotide is having this free group again. Hey, free. The one that is even this nucleotide. Hey, it's having this free. So what happens? If you want to form a polynucleotide, now those free groups still do the same. You bring another nucleotide, and then things keep on happening and happening. What you're not supposed to forget is that now we start dealing with carbon number 3. So why carbon number 3 had the OH projecting downwards is because that's how we are going to form the polynucleotide. That's why even the chains always just go down, because now we are going to come here and bring another nucleotide. So when you bring here another nucleotide, guess what? You'll be forming now a tri. So when you bring there another nucleotide, you form what we call a trinucleotide. Now, after getting a trinucleotide, and then you bring another nucleotide again. So trust me, to also have that free group down, you try it and see. It will try, sorry, it will do the needful. Remember, when you bring the nucleotide here, it will have a free OH here. You understand? And then here it will have the things of phosphorus. You get, it will have the P in one way or the other like that. So that means it will also remain with this free thing here. So when it comes, it will have the what? The OH here. Then when you bring another nucleotide, this nucleotide will have the P, and the P will also have the what? The OH. But remember, the one you brought, it's also having what? A free OH. That's how things keep on accumulating and accumulating. I've started losing my voice. Hey. Let me hope you people have gotten that. So the trinucleotide, after forming the di1, you bring a new nucleotide. I've said it will have this one projecting down, and then the phosphate group. More condensation happens, you form the tri. You bring another nucleotide, it will have what? A free OH and also this phosphate group. You get it? So they will have interaction with only the OH of the trinucleotide, which came from here. And things will just be like that, like that, like that, like that. So have it in mind, for you to form longer chains, it is always what? One of the free groups on the phosphate group and the free OH group on carbon number 3, just like the way this thing says here, this statement. It's saying a free OH of the phosphoric acid, but don't be surprised when it's called the phosphate group. Different textbooks write different things, but of course they are all okay. They're all okay. Yeah. But I think phosphate group would be better. So I think you will understand if I do that. Yeah. Phosphate group, phosphoric acid. So I want people to tell me where exactly is the problem. Or in simple terms, can you form the polynucleotide? That's what I'm asking. Do you get finally how we form the dinucleotide, and can you form the polynucleotide on your own? A polynucleotide simply means add on more 2 nucleotides and you've reached. Sometimes they even tell you a trinucleotide is a poly one. So are we together there, people? Are we together there? Phosphoester bond different from phosphodiester. Can you draw those structures? Because now we are going to reading the statements and then we leave it at that. We leave it at that. So we are having our next lesson. Let me see the timetable. Where is the timetable? Where is the timetable? Who can tell me when exactly is the next lesson of biology? According to the timetable. When is it? When is it? I know someone knows. Yes, Nicholas Owen. Is it tomorrow morning?
Yes, this tomorrow morning.
Okay, thank you. Yeah. Tomorrow morning is not very far. Tomorrow morning is not very far. So tomorrow morning we are going to begin with watching the videos as you recall. So as we watch the videos, I will ask members to go back and review the structures of the polynucleotides. Then we do the structure of DNA and so on and so forth. So what are people saying in the chat? Can you do the polynucleotide on your own? You people, can you draw the polynucleotide on your own? Simply adding on more 2 nucleotides and the same things will be happening. Yes, condensation between the free OH on carbon number 3 and one of the free OH on the phosphoric what? Phosphoric acid. So the assignment is to show the formation of a polynucleotide. Show formation of a polynucleotide. Yeah. Yeah, you first, sure. Don't bother yourself with the statements for now, but the statements are very important. First, sure. Then you can even make a write-up. But I think you're going to see the write-up in my notes. But please don't cheat. Don't cheat. Don't cheat. Do the needful as I've asked. Okay. So very first, let's go through these notes. Very fast. I think most of the things have been talked about. Yeah, we are not going to hover so. Yeah. Yeah. So this is what I told you, that in DNA, this D is for deoxyribose. Then I told you that this Na is for nucleic acid. So as we write the full word, we remove this. Yeah, just say deoxyribonucleic acid. Yeah, deoxyribonucleic acid. The same with RNA. So for RNA, let me just write it. So for RNA, instead of saying ribose, you just say ribonucleic acid. Ribonucleic acid. Then the other one is deoxyribonucleic acid. This one is ribonucleic acid. RNA. Then this one will be DNA. But how do you write it? Deoxyribonucleic acid. And what is that? DNA. Simple as that. Okay, let's see what we have here. So they are saying, so very fast. Now this is like a review. A nucleotide has 3 compounds, 5 carbon, sugar, nitrogenous base, phosphoric acid. Make sure you understand these statements. What are they saying? The sugar has 5 carbon atoms, therefore it is a pentose. We know that. Nucleic acids containing ribose, as they are pentose sugar, they are called ribonucleic acids. All those containing deoxyribose. What is deoxyribose? It is ribose with an oxygen atom removed from carbon number 2. And they are called DNA, ribonucleic acid. See that? Very okay statement. So we came to structure of a nucleotide. You can see these two OHs are pointing up, and they are telling you at carbon number 5, that's where we join to the phosphate, or I said phosphoric acid. Then carbon number 1 here is where we join to the what? To the nitrogenous base. We know the reasons. And from carbon number 2, that's where we remove oxygen to form deoxyribose. But since the oxygen is here, this is the structure of what? Ribose. Are we together? So we go. Purines. So they're saying purines, 2 rings, and I told you one ring is hexagonal, the other one is pentagonal, as you can see. And you all know they join to the sugar at those points, but don't mind about them. They shouldn't bother you. Then pyrimidine, it's only 1 ring, and it is what? Hexagonal. Hope you know that. Phosphoric acid, you can see the structure. That is it. And they are telling you that this is the OH that will join to the sugar. By the way, it's a many, so any can work. Are we following? So it's like a review. What are they saying? Bases, each nucleic acid has 4 of them. We are going to see this in the structure of DNA. Yeah. Right now, we may not need it. Has 4 different bases. 2 derived from purine, the 2-ring bases, and 2 derived from pyrimidine, the 1-ring bases. You get? So they are telling you that the nitrogen in the ring gives the bases their basic nature. Of course, you know, without nitrogen, we cannot have amines, and without amines, we can't have basicity. So they're telling you the purines are 2, gapurine, guanine, adenine, G and A, pyrimidines, thymine, and cytosine. Then we also have uracil, as I said. These things they are saying of RNA and DNA, we shall see those tomorrow. Remember I told you uracil has bad manners. That's why it doesn't resemble the names of the others. The others end in "-ine", or it ends in "-il". And you see that it is very weird. It is not even there in DNA. Yeah, it's not there in DNA. That's why we call it something with bad manners. But we shall see that as we discuss the structure of DNA. So they are saying phosphoric acid gives nucleic acids their what? Their acid character. We said that, I think. So they came to formation of the nucleotide chain, or just a nucleotide. I think you can see the phosphoric acid undergoing condensation at carbon number 5. Carbon number what? Number 5. Yeah, there is something I forgot, and I'm going to talk about it, don't worry. And I think you can see that at carbon number 1, we have another condensation with the nitrogenous base. So don't mind these structures. The structure of the nitrogenous base shouldn't bother at your level. It shouldn't bother you. So you just break a box, put nitrogenous base. Expose one of the hydrogens, show how condensation is occurring. Just actually consider my structures, they are the same. I think you can see here the phosphodiester, sorry, this is phosphoester, because condensation is only happening here once. Yeah, don't be like, but it's also happening here. No, the condensation here gave us the covalent bond between carbon and nitrogen, if you can hope we are together there. So here they are saying the sugar is deoxyribose because you can start carbon number 2, it is H not OH. That means the H was lost. Hope you people know why with formation of the nucleotide we lose 2 water molecules. Because we have condensation happening twice, forming the phosphoester bond and the covalent bond. Or we have condensation at carbon 1 and carbon 5. That's why we lose 2 water molecules. We got formation of the polynucleotide chain. You can see the same thing has been happening. You see, they got the OH of the phosphate group, and condensation happened with this OH on the carbon number 3. And then after that, and these people have the dinucleotide. It's not even the polynucleotide, it is the di one. But I told you the same things are done. That's why they said poly, but they showed di. You get it? Yes. So you can see condensation is happening, so we form phosphodiester bond, and I told you why it is phosphodiester bond. You can see now, do you see that the bases they're just putting there the word base because you don't need how they look like, and you should just make sure that they are projecting at an angle of what? At an angle of 90. Projecting at an angle of what? 90. Nothing much. So here they talked of uses of the nucleotides. They say they are building blocks of nucleic acid. That's what we began with. Then them being present in coenzymes, we shall see this later when we see their nomenclature. Yeah. You will see that this D stands for dinucleotide. You will see that this one stands for triphosphate. You see what they mean with time later on. So as of now, they shouldn't stress you. But you've met them in nutrition and respiration. But I will repeat them, maybe as we revise. So I think you can see that formation of a dinucleotide alone can represent the formation of the whole polynucleotide. Okay, what are they saying? 2 nucleotides join to form a dinucleotide by condensation between the phosphate group of one nucleotide. Are you seeing? Here they are not even saying the OH of the phosphate group, they are just saying the phosphate group because the phosphate group has the OH with the sugar of the other. You see, they're not even saying with the hydroxyl group on carbon number 3 of the sugar, they're just saying sugar of the other. They are not even saying OH group on the phosphate group, they're just saying phosphate group. So you should be a good biology student to understand why they summarized. So they are saying that this process is repeated several times. That's why I told you that the same thing is happening. So they repeat it several times, and here they are saying about 1 million times to make the polynucleotide. You understand? And then what is formed? What is formed? They called it unbranched sugar phosphate backbone. Someone may be like, what do they mean by the phosphate sugar backbone? Look here. You look at what we had. Look at the structure that we are coming up with. we are coming up with what we have here is a sugar. Are you seeing this is a sugar? This is a sugar. Then what you have here, the whole of this is what they call a phosphate group. Yeah? Phosphate group. You people, you learn what they call you learn these things with time. You all know that the phosphate groups looks like this in chemistry. It has the P and O oxygens. I told you that in chemical formula in oxygen. So you can see the P and the 4 oxygen. So the phosphate group is there in one way or the other. So that's why they are calling it the sugar phosphate backbone. Yeah? So you can see that as this structure of ours becomes longer and longer, as we go down and go down and go down, what shall we have? We shall have a sugar phosphate backbone. Sugar phosphate, phosphate, sugar, sugar phosphate. I don't want any statement to confuse you as you're reading. So hope you've seen how the sugar phosphate backbone comes about. How it comes about. Now, let me see. I think we are even done. Yeah. So what we are starting from, or our next thing shall be the structure of DNA. We shall watch the videos tomorrow, but you're going to go to YouTube and watch them on your own. Then when we come, so that will be very fast. So I want someone to go to the discussion group right now and tell those people that doctor said, go to YouTube and watch the structures of DNA and all forms of RNA. Yes. So let me give you this last thing, which is food for thought. It is not so important at your level, but it is good to know. It is good to know. It is good to know. Good to what? Good to know. Good to know. So, remember, we have a pentose sugar here, not so? We all know that this pentose sugar is touching a nitrogenous base here. I want to tell you something important. Good to know, nitrogenous base. Then we know that here we are having the phosphate group. You know the phosphate group is formed after condensation with phosphoric acid. And the phosphate group. Or people say phosphoric acid like that. Who can tell me what is the name of that structure? What you're seeing on the screen. If I combine all those things, what is the name of that structure? What are people saying in the chat? Nucleotide. Thank you so much. Thank you so much. So, people are saying that what you have here is a nucleotide. So I'll come here and say structure of a nucleotide. Now, someone tell me, what is the name of this thing? Only this part. This part has a name. That part has a name. What is the name of that thing alone? Yes. What's the name of that thing alone? Huh? Nucleoside. How? That person is reading. That person is reading. That is what they call a nucleoside. But they will not examine it. It's just good to know. That thing is what they call a nucleoside. A nucleoside. Yeah. So, am I right to say, therefore, so you can brag. I'm even going to pinch and you put it on your status. You can tell people that a nucleotide is a combination of a nucleoside. Remember, nucleoside is a pentose sugar with a nitrogenous base. Is a combination of a nucleoside plus the phosphate group. Trust me, very few students know this in Uganda. Yeah. Yeah. Very good to know. Let me pinch it. Yeah. That is something you can brag about as a biology student. Yeah. Pentose sugar, nitrogenous base, they form a nucleoside. You add the phosphate group, that one becomes a nucleotide. Good to know. Good to know. Very good. Take a screenshot and post it. Okay, this is my assignment. This is my assignment. My assignment is to watch, though I'll bring them in lesson 2, but you're supposed to also watch them. And I also want you to be sharing them in the discussion group here. Every YouTube video you find, you share the link in the group. You share so many so that people watch. So that by the time we come here, we are very good. So you can watch. You watch YouTube videos. YouTube is capital I, capital T. You watch YouTube videos on what? On the structure DNA. On then on DNA replication. Don't forget it. Then on the structure of all forms of mRNA, of RNA, sorry. So let me even help you with research. You may ask, what are those forms? The forms of RNA at your level, the ones we study. We study messenger RNA. You study tRNA. Then you study rRNA. So you can watch those YouTube videos. Trust me, when you come back for the lesson, it will be very easy for us. So I think this is the end. Yeah, that's the end of the session. I've introduced nucleic acids. So that means next time we are now going into the deep content. So the videos will be like reading ahead. So every biology lesson, I always leave things you're supposed to read so that when you come, you read ahead. You're also covering this lesson ahead of your teachers like that. And that's how you will pass. So allow me sign out. Allow me sign out as Paul Tutor, a tutor of math and science at Science Scholars Academy based in Uganda. eye contact 0 point, sorry, it is 70, not 7, not 0 point, it is 075327 95 92. I prefer WhatsApp calls. You call until I pick, even if it means calling for more than 4 days. Yeah. Yeah, that's it, people. That's it. So our next stop is what you're supposed to watch on YouTube. So I'm supposed to share this workbook to the premium group immediately after the what? The class. So we are going to have our very first biology drive. Yeah. So we are going to have our very first biology drive where we are going to be putting most of the biology work. And then as well, most of our work next year, it will be found on our app. I'll tell you the name of the app in the future. Yeah, but most of our work will be found on our app. I'll tell you the name of the app in the future. So that because people have issues with visiting the Google Drive, but that will be in the future, where our work will just be found on our app. You can download it and then access the work. Just wish us well. Otherwise, thanks for attending. Let me check the chat and I can rest. Thank you, sir. Thank you, doctor. Thank you so much. Thanks, doctor. Great tutor. Thank you. Okay. Thanks for appreciating too. You're welcome. Till we meet. Bye-bye.